Yes, it's working. All right, welcome back everyone today on the second day of the G by E by M symposium. We're going to start the day off with a session sponsored by Corteva, which I and some other PhD students from Wageningen had the pleasure of organizing. So, I just want to thank Eike, Hugo, Junita, and Jonathan, and Yingye for organizing this with me. I think we have a nice list of speakers. We have two sessions. We're starting off with some speakers who will talk about AI in G by E by M. And then after the break, we're going to have a second session on, well, let's say more diverse aspects of G by E by M, which might not normally show up at a conference like this. There has been a slight change in the schedule because we were under the impression that Jonathan was actually in Europe, but turns out he's in Iowa where it's currently 3 a.m. So we figured it might not be a very good idea to have him present at 3 a.m. So he has swapped with Eike. Eike will be presenting, and then Jonathan will be giving a talk later this afternoon. So the idea, I think, is that we have 15-minute talks, 5 of them, and then there's 15 minutes, I believe, for a discussion at the end. So, if there's a short question after the individual talks, I think that should be fine. But let's look at the time and just remember that there's always room for questions during that discussion session. So I think we can get started then with Eike presenting on AI in G by E by M.
Okay, good morning everyone. I'm happy to be opening this AI session with some of my work. I am Eike Potze. I'm a PhD student at the new WUR AI group, and I also work at Biometris with Fred, of course. And my goal in my PhD to make deep learning work for the problem that many of us care about, which is the prediction of G by E by M. But because I'm opening this session, I also want to give a bit of a broader context of AI research for different problems. Because what we're seeing in AI research for, I think, modeling complex systems is we're seeing many breakthroughs, but we're also seeing a lot of care being taken to make sure the results are right. I think this is the right approach. So we need to be optimistic regarding AI research, because it can really solve big problems that we have. But we also need to be critical to make sure that the results really are what we're thinking they are. So let's get to G by E by M prediction. So what is really the current state of G by E by M prediction? And to answer this question, we need to look at benchmarks. Because in benchmark datasets, you can compare different classes of models on the same dataset and give an honest and unbiased comparison, you'd say. So the biggest and, I think the best benchmark for a G by E by M prediction nowadays is the Genomes to Fields dataset, because it's very large. So it's a multi-environment trial made by our American colleagues by aggregating a lot of maize trials across the US. So you have a lot of trials, 250 environments, 5,000 maize hybrids, I think around 140,000 yields in total. So really a big data set where you can test your methods on. It's fully open, everyone can use it. So we're really sure we're comparing against all the different methods. So the challenge is quite big. You want to predict for a new year and for completely new maize hybrids, which haven't been tested. They do repeat the locations though. You have all the features you would want for a deep learning model to succeed. So you have your markers, you have your weather time series, soil measurements, and management features. So great, sounds great, right? We have a hard problem, big data set. Let's see how deep learning does for this problem. Well, it turned out it didn't really do that well. So the winning methods to the right are some combination of machine learning methods, classical machine learning methods, and linear mixed models. The winning team is actually a combination of both. And deep learning methods stayed quite a bit behind. So the best deep learning method in this competition was number 7. When we look at the predictions within specific environments, so the correlation within environments, it's actually worse. So it really looks like deep learning models currently are not doing well at this. And this is surprising because you'd think, okay, these are nonlinear relationships you're learning, you have enough data to do it, so why isn't it happening? So that's the question I'm starting at, why? So we have enough data, why are deep learning models not succeeding? Do we need 1 million plots or 10 million plots before we start to see an advantage? That would be ridiculous, I think. So the reason I want to point to is that deep learning models are prone to learning shortcuts. So this is a well-studied phenomenon. Like you can show it, for example, if you want to detect cows. But what turns out that usually the model, if you have a small data set, it usually learns to detect grass instead of the cows. So if you have a cow on the beach, it doesn't see the cow, it just doesn't know what to do. So because these cows and the grass are dependent in the samples you have, the model goes for the easiest pattern to learn, which is the grass, and it doesn't actually look at the cow. Okay, that's a well-studied phenomenon. And then when we relate this to multi-environment trial data, you can think about it in this way. So if we do a very simple two-way ANOVA based on the different factors in your design, so your genotypes, environment, and your G-by-E interaction, you see that usually the environment is repeated many times, and it explains most of the variance. So what this means is that there's a really strong signal for the model to pick up on, and it's also repeated many times. So usually the samples for deep learning models are sampled independently, but here it's many repeats of the same pattern. So what we can expect from this, okay. The clicker's not working. Okay, great. So we can expect the optimization of these models to be biased towards learning the environmental effects. That was also what was shown in the rankings. So if you look within the trials, look at what you're predicting the rankings, you're really not doing that well. And we address this with a very simple conceptual approach. So we try to correct this imbalance by using the design of the multi-environment trial and with a standard linear mixed model. Okay. So we start by decomposing the targets we have for a deep learning model with a normal linear mixed model. So what we do, we don't treat all of these plot yields as independent samples. We say, okay, they're actually samples of genotypes and environments, and we formulate a mixed model to decompose this structure. So we decompose the environment by location and year as well, but this is sort of I guess, almost the minimum linear mixed model you can formulate to decompose this variation. We take out the locations because we know the locations are repeated, so you can actually directly estimate their effect instead of predicting it. Okay, next we obtain the blobs for the environment term, the genotype term, and the interaction term. And we use these to train individual deep learning models. This is kind of an unusual approach, but this means that we can actually decompose the big problem of predicting yield in the next year into 3 independent problems. And the first of which is very well known, just genomic prediction. You have your markers, a deep learning model, and your blobs, so you are able to predict this for the next year. Then the next one is also a well-known problem, yield forecasting. You have your weather, your soil, and your management, and you just try to predict the trial means. And on top of this, we train a model which reuses the embeddings of the earlier models to predict the interactions. And if we separate this problem in 3 components, it's a bit easier to train these models, and they're not distracted by the signals of the other models. So when we combine these predictions, we substitute them into the linear mixed model formulation, and then we reconstruct the yields that we're interested in the next year for new hybrids. Okay? It's conceptually, I think, it's quite reasonable. So let's look a bit under the hood on how we actually train these neural networks or how we develop them. Because the problem we have is that there's many options to choose from when it comes to these neural networks, and we need to tune them very well. So we have a lot of candidate models, you could say, and we need to develop 3 individual models from them. So what we do is we have some sort of funnel, you could say, or a pipeline to develop these models. We start with a lot of model configurations which we test once, and they're cheap to train because we only, for example, have 250 trials to predict, so they're actually quite quick to train. Then slowly we narrow down the options and choosing reliable hyperparameters for the best model. And then finally we reinitialize the model until we get a stable model. So this kind of pipeline allows us to go a bit from exploration of individual models from literature to actually getting a really well-trained, reliable model. And I want to give you a bit more of a look under the hood in the results and how the individual models are doing at predicting these variance components. Oh, Yes. Before that, also say we're going through this pipeline 3 times because we have 3 modeling problems and we are developing 3 individual models. Okay, so we do it within the training sets, we do an 8-fold cross-validation. We fold out part of the data, train a model, and see how it's doing. So what we can see is that these are really 3 independent modeling problems. So actually, you have a lot of examples for your genomic main effect, you have a lot of examples for your interactions. So you can see that there's quite a low variance in these predictions. But for your predicting your trial means it really matters what kind of validation set you select. So you really need to select your models there for reliability, actually. So the prediction of each of these components is its own modeling problem. But if we decompose the prediction problem into these 3 components, we actually have a sort of divide and conquer approach. So you can solve them separately, and you can use methods from literature to optimize them separately. So if you combine these models back again and compare it with the competition, then you get the full yield predictions. And I just want to remind you, this is in a new year for new hybrids. And we're actually doing really well. So it looks like the problem is, with this kind of approach, you address the biggest problem already. Even with a simple linear mixed model, you sort of fix this imbalance with optimization, and we're comfortably in yellow ahead of the existing deep learning methods. And the same pattern holds true when you look at the within trial. So this is actually the biggest issue we wanted to solve. You're actually predicting the yields of individual genotypes in individual trials, which is what we're interested in for genomic prediction under G/E. And we actually see that it looks like we mostly solve the problems with deep learning models for this approach. Okay. So in short, this target decomposition that we do, in general, it improves deep learning performance in these kind of yield prediction problems. So I want to end with 3 take-home messages based on this short research talk. So the first one is, I think, obvious to the statisticians, but deep learning methods really shouldn't ignore the design of your multi-environment trials. So not a randomly sampled observational data set. There is a really clear thinking that goes into this design, and you should use it, because if you don't use it, it might still influence your predictions. And it's also something that statisticians and deep learning researchers could really discuss and share experiences about. So the second part of this kind of prediction problem is actually a collaborative effort with many components that can all be improved. So if you have a better design, and if you are better able to model your G/E with a linear mixed model, if you have better genomic prediction models, all of these things can contribute to the prediction of interactions. And we expect with all of these different research problems that this kind of approach will get better over time. So it's actually something that many people can contribute to. The only part of this that's actually really hard at the moment to fix is the interaction prediction, because you really need all of these components in place before you can even start researching this. And I think that's why I want to end with one final take-home message, is that we really need more and better benchmarks for this problem. Because I think AI research in general is organized around these benchmarks. And if you want to have any sort of large progress in AI, you really need some sort of data set in place that everyone can play with, everyone can compare with, and that you can develop targeted methods for. And I think especially for the interaction prediction part, we really need these kind of data sets to make some real progress as a community. So that's something else I want you to think about, like what would this data set look like? What kind of metrics would really be which represent the problem that you actually have? Because I think all of us have a different view of this problem, have a different maybe multi-environment trial maybe, but I think there is some shared problem that if it's solved by an AI researcher that doesn't even look at the trials individually, but if the benchmark is solved, it would solve your problem as well. I think there's a real opportunity there as a community to define such a benchmark and enable some sort of long-term AI research in this space. Okay, that's what I want to end with, and thank you very much for listening.
All right, thank you very much, Eike. I think we have time for one quick question before we move on to the next talk. So yes, a question there. Oh. Where's Felix? Yeah. Oh yeah, I mean, if you want to, you did quite a good job yesterday, I think. So. Over there.
Thanks a lot. Thanks for this good presentation. It's very interesting you like split up in components, but for the environmental component, we've seen yesterday as well that it's very difficult to have lots of multi-environment trials, so you don't have much data. Like, have you considered for including mechanistical knowledge, like including a crop model on the environmental component and then going on with deep learning, like to integrate it in, like just like include mechanistical knowledge inside that environmental component?
Yeah, I agree that this would be a valuable way to solve it, especially with small data. But I think it also relates to decomposing the problem. So this is actually a really active research area in general within yield forecasting, and you can switch out your yield forecasting model within this framework for a hybrid model without any problems actually. So I think if you have a small data set, this is a very valid option to use this kind of model. But I think even there you need to worry about sort of the structure of your data, because if you only sample, for example, very few locations, then it's really hard to predict for new locations. Like, so even there you have to worry about these kind of the underlying design actually.
Yeah, for example, I'm thinking about like weather-soil interactions, because like weather is definitely, or like soil is definitely affecting the way how plants can have access to water. But it's very difficult to have these in like just the neural network thing, because you need there's lots of interactions there. Like maybe it could help like just elucidate the systems or the mechanisms